Hello, everybody. We're just going to give it a minute or so for everybody to join. See, the number is still going up, so I'm just going to wait another couple seconds. Good. So welcome, everybody. I'm Merit Cudkowicz. I'm joined here by a number of people. We have an hour session today where we're going to give an update, as always, about the platform trial, and then also spend some time talking about the recent announcement of the Rescue Clene study from Australia. And we have with us today Michael and Robert, who I'll introduce more officially later, from Clene Nanomedicine, and also Nick Maragakis and James Berry, who co-lead the Regimen C for the platform trial. And then we have Laurie Chibnik, one of the MGH biostatisticians, who's going to give us our first kind of lesson on biostatistics. So for those of you who are new, this is our weekly webinar about the Healy platform trial. This is really a community-based effort to increase how we, the speed of testing new treatments in ALS. And you can see by all the logos here how collaborative it is, both in the funding and the conduct of the study. Next slide, please. So just briefly, this is a perpetual adaptive trial. That means that we keep going until we find effective treatments. We've started with 4 regimens. We call them A, B, C, D. The names of the drugs are here on the right. And we have, many of you know, taped science seminars about each of them that are found on the Healy ALS Center website, if you want to know more about their science. It's a 24-week study where 75% of people get active drug of one of these 4 treatments and 25% placebo. But at the end of the analysis, we compare everybody on active drug to the shared placebo. And that's where you get a lot of the economy of time costs. And it's much more patient-centered. And then when we want to add a drug, we can just amend the program, the protocol, and that's much faster than starting a trial on its own. After 24 weeks, everybody has an option for open label extension. Next slide, please. So as we've been asked, we give our update every week on where we are in enrollment. I'm very happy to share that we are nearly done with enrolling for A, B, and C. You can see the numbers there, 160, 167, and 161. We have one person that is in screening and about to go into regimen A, and when that person's randomized to A, we are going to be officially done enrolling in regimens A, B, and C. So that should be soon. And we're not that far behind in finishing enrollment in D. And we're also working on adding the 5th regimen. So this is really good news. And we'll definitely share when we hit that really important milestone, because that sets the time of when we're going to have the results of these first 4 treatments in the platform trial. Next slide, please. So I wanted to now formally introduce our guest speakers, Dr. Robert Glanzman, Chief Medical Officer from Clene Nanomedicine, Michael Hotchkin, Chief Development Officer from Clene Nanomedicine. They're going to speak first. We asked them to come and to explain the results of their Australia study with Clene. And this, again, is our Regimen C in the platform trial. And then afterwards, Dr. Laurie Chibnik, who's an assistant professor and biostatistician at the Harvard School of Public Health and Mass General Hospital, will give us her first lesson. So with that, I might pass. Oh, yeah, one more slide, sorry. So each of our regimens has co-leaders who are really responsible for leading the trial. And for Regimen C, or the Clene Nanomedicine regimen in the platform trial, it's Dr. James Berry from Mass General Hospital and Dr. Nicholas Maragakis from Johns Hopkins University. They're also here to talk about the Clene study. So with that, I'll turn the floor to Michael and Robert, perhaps to start.
Thanks, Merit. This is Robert. I'm having some technical difficulties, so I can't share video, so I apologize about that. So let me just give you some background. I don't know if the participants here on the call have heard about our press release regarding Rescue ALS, which was a phase 2 randomized double-blind placebo-controlled trial of CNM-Au8 in early ALS patients that was run at 2 centers in Sydney, Australia, using a really novel primary endpoint called MUNIX, or Motor Unit Index. which is an electrophysiology measure of low motor neuron preservation. So we reported negative top-line results. We did not meet statistical significance, although there was a benefit of CNM-Au8. It just wasn't significant in the ITT population. And let me just explain, well, that was really because we enrolled patients both with limb onset and bulbar onset, who are sporadic ALS patients, and the bulbar onset subgroup simply didn't progress. So that diluted the therapeutic effect of CNM-Au8, which was seen in the limb onset population who did progress. And we saw a significant therapeutic effect at week 12 with a very strong trend at week 36 with a p-value less than 0.08. So in terms of the electrophysiology endpoints, again, we saw statistical, I would argue statistical significance in the limb onset population, but not in the bulbar onset population who really didn't progress at all. But in terms of the key secondary endpoint, forced vital capacity, again, we see evidence of directional therapeutic gain with CNM-Au8, approximately a 20% relative improvement versus placebo. But again, this wasn't statistically significant. And of course, the study really wasn't powered to look at this. There were also some important exploratory endpoints, including quality of life endpoints, as well as the proportion of patients who had a 6-point or greater decrease in ALSFRS, and as well as disease progression, which was defined as reaching King's clinical stage 4 or death. And all of those endpoints were statistically significant in favor of CNM-Au8. But I think, as optimistic as we are about these results, they need to be put in context because no adjustments were made for multiplicity. So I think from our perspective, at least, we do feel that this study clearly showed proof of concept for CNM-Au8, but of course, confirmation of efficacy needs to be confirmed in a larger trial like Healy. So we're very, very gratified to be part of the Healy program. In terms of safety, CNM-Au8 has been shown to be extremely safe in all of our clinical development programs to date. We have over 300 patient years of safety data and growing, and we have not seen a single serious adverse event related to CNM-Au8. And of course, the drug seems to be extremely well tolerated as well, because we really have not seen discontinuations due to tolerability with CNM-Au8. So I think that pretty much summarizes the way we view the data, at least. And we hope that that provides some perspective, because I think some of the participants here may have heard a bit of a different story.
Michael, would you like to add anything to that? And thank you, Robert.
Certainly, thank you, Merit. I think one point that I'd add to Robert's very eloquent discussion of the results is how excited we are to potentially be bringing a treatment to the community. We think these early results are early, but really indicate or point to the possibility of benefit that we'll learn and understand through the Healy platform trial. And we also think those results are important because it begins to signal that part of the pathophysiology of ALS may be related to this, energetic impairment, which we hope to address with CNM-Au8 as a treatment.
Thank you. So please, if people have questions for anyone on the Zoom, please put them in. But before we go to, we're starting to see a few. Before we go there, I just wanted to see if Nick or James wanted to add any of their thoughts.
Well, thanks, Michael and Robert. That was actually, I think, really helpful. I think the thing that we would emphasize with regard to the Healy trial is that it is going to be a larger trial. The Rescue ALS study was about 45 patients, and of course, the Healy will be 120 participants who are on active drug, and then a sizable placebo, shared placebo population as well. So we think that this will also get us to a statistically, and you're going to learn about biostatistics today, will give us hopefully a cleaner answer. And I think the trends were certainly interesting, as both pointed out. So I think this larger group will be helpful. I want to point out for participants as well that we're using 2 different doses of the drug. The 30 milligram dose is the lower dose that was used in the Rescue ALS study that they just spoke about. But in addition, Healy is using both the 30 and a higher 60 milligram dose. So I think that will provide us information with regard to potential efficacy. It'll also provide us information to their points with regard to safety. And then, of course, this open label extension, which is part of the Healy trial, is different from the Rescue ALS and potentially could give us information about long-term efficacy and safety. I also want to emphasize that the platform trial has frequent futility stopping rules and investigators who are overseeing that. So if there are any concerns about safety or even efficacy, the trial will be stopped and those data analyzed. So we've enjoyed working in cooperation with this group, and I think through the Healy platform study, we'll get an answer.
So we do have some questions coming in, so maybe I'll read them and maybe suggest some people to answer, but anyone can answer. So Robert, maybe you could take the first one. If you could please repeat which group benefited, limb or bulbar, and if you know what % were fast and slow progressors. And maybe you could also qualify the word benefit here, given the design of the study.
Sure, thanks, Merit. So again, in terms of the primary endpoint, which was an electrophysiology measure of an estimation of low motor neuron dropout in the limbs, we studied 2 muscles in the hand, a muscle in the arm, and a muscle in the leg. In terms of that primary endpoint called Munich's, the limb onset patients benefited. And when I say benefited, they did better than placebo. So there was a statistically significant benefit at week 12 and a strong trend at week 36, again, with a p-value of less than 0.08. And that was only in 33 people. So we view that as strong evidence of neuronal preservation in the spinal cord of patients with limb onset ALS in this study. And of course, the bulbar, this is an early ALS population. The mean time since symptom onset in this population was around 16 months. So they were fairly early, and the bulbar patients simply, their spinal cords apparently were relatively normal because they really didn't progress in the context of the 36-week study. So I think that hopefully that puts the data in perspective.
And Robert, you might add some context on the effects in bulbar patients for the clinical endpoints.
Yeah, I think for me, that's the most critical thing. When you look at the actual hard endpoints, especially disease progression, again, as defined as reaching King's clinical stage 4 or death, it was the bulbar patients that really drove the benefit seen with CNM-Au8. When we talk about a benefit, we're talking about a 67% relative risk reduction in reaching King's clinical stage 4 death in that population in the study. So we're very encouraged by the data. We really feel like these clinical endpoints, although, again, the study, we didn't make any adjustments for multiplicity. On the other hand, there wasn't a single efficacy endpoint that was in favor of placebo seen in this study. They were all in favor of CNM-Au8, although not all of them were statistically significant. So again, we are very encouraged by the data. We're very gratified and happy to be a part of the HEALEY ALS Platform Trial. And we agree that this trial should hopefully give us an answer.
I wanted to maybe expand on the bulbar, because we do have some questions that I think are misunderstanding. So I think this is the time to explain. Someone asked, is it true that the drug did not help bulbar? And I think it's important to say that we don't know that. It's just because the test, the Munich's, is only a test we can do in the limb muscles. We can't do it in the bulbar muscles. And these participants were very early. They didn't have abnormalities of that measure in their arms, either the treated group or the placebo. So you really couldn't determine for the bulbar onset participants if there was a change in Munich's. So we don't know really on that outcome measure for those bulbar onset. So I don't think, we don't want people to think it doesn't work in bulbar, because we really don't know. That's one important thing. I don't know, Nick or James, if you want to add to that.
Yeah, I'm sorry, I'm not on video. But I agree. I mean, I think the takeaway is just that our outcome measure limited our ability to get information on people who had bulbar disease. Not that the drug has any differential effect. That's really the key point to take away from that.
That was said much more eloquently. Thank you.
Merit, this is Robert. If I could just add, again, in terms of the clinical endpoints, it was a robust benefit of CNM-Au8 in bulbar onset patients. So in terms of hard disease progression, we had a 67% relative reduction in risk for reaching those hard endpoints in the bulbar population with CNM-Au8, which was statistically significant.
Thank you.
Yeah, I think that dovetails potentially into one of the other questions, and that asked about endpoints. And to that point, the ALS FRS, which is the endpoint for the primary endpoint, at least for the platform, will incorporate, as many of you know, will incorporate many aspects of ALS function in participants. So hopefully, again, to this point, we'll hopefully get a broader signal that will include bulbar ALS patients and their dysfunction or their limitations, as well as limb onset.
Thank you. I think it's probably a good time maybe to ask Laurie just a spot question about just the small sample size. I mean, I think this is a really important study and it confirms our interest in proceeding with this drug in the platform trial, but there are some limitations to interpreting small studies. Do you want to weigh in a little bit on that?
Sure, absolutely. And I see there's a question in the Q&A that kind of falls under that as well, with the subgroup analysis. So yeah, it's always quote unquote easier to see differences when you have a large N or a large amount of people in the study. And this is kind of analogous to if you flip a coin. If you flip a coin 3 times, the likelihood that you'll get 3 heads doesn't make you think that the coin isn't a fair coin, because 3 is just not enough flips. But if you flip it 100 times and get 80 heads, that makes you think the coin might not be a fair coin, right? So it's the same sort of thing. A trial of 45 patients is still pretty small. And I believe that's the number that was in this trial.
Yes. Yes.
And then when you start thinking about subgroup analyses, that number gets smaller and smaller. So it just makes us a lot less confident that what we see is a real difference. It's a lot harder to see that difference because that signal or that difference has to be bigger than the noise. Fewer people in the study, you have more variability. So that's one of the issues. So with the Healy study with 160 participants, which is 3.5 times bigger than this study, we have a lot more power, is the word we like to use, to see a significant difference. So I think the results from the Healy are going to make a really big difference on our understanding on whether this drug works.
Thank you. So there's a question here. The CNM-Au8 has been used in the EAP for a good amount of time as well. Do we have any results from the EAP that are significant? And I might take that because I think there's 2 EAPs going. And again, I'm very thankful to Michael and Robert for making that an option for people with ALS not eligible for the platform trial. We don't have results yet. I would say that there's no safety signal at all. Because we don't have a concurrent comparison group, like a placebo group. It's not as straightforward to look, but we are working. We're planning to work with Michael and Robert and our biostatistician consultants and colleagues to see if we can compare people to PROACT and try to learn something more from that. But at the moment, we haven't done those analyses. So the other question, let's see, and we answered some of it, but maybe Nick, you can answer. Are the endpoints the same in the rescue ALS and the Healy trials?
Right. So as pointed out, the rescue ALS was the use of Munich's as the primary outcome measure, which is this nice electrophysiological measure. And for the platform trial, it's the ALS FRS, which is a much more kind of global scale. And so different endpoints, both I think interesting and both helpful.
Thank you. Okay. So there's a comment that I think for all people with ALS, they'd be grateful for a 20% difference in breathing score, and thank you. This is kind of related to the subgroup question. Will you be releasing data at the subgroup level? Will the N be high enough? I don't know if Michael or Robert, you want to talk about the data release and your thoughts on this.
Sure. So there'll be some data will be presented at the upcoming ALS MND conference in December, and we do have some subsets broken out for the slowing of the disease progression endpoint by Bulbar and limb onset, which is what Robert's been commenting on, and then we'll also display the top line Munich's data that Dr. Maragakis just commented on. For the limb onset and the full population. So yes, some of those data will be presented shortly.
Okay. Related to that question, there's a question, given the top line difference in response noted between Bulbar and limb onset, could you share or show that data so we can see what you're talking about? And I'll maybe just answer the why we're not showing it today. So anytime we're presenting to people who might be in the trial or want to be in the trial, we do need those slides approved by our ethics committee. And we just have not obviously had time since this announcement to do that, but we still wanted Robert and Michael to talk to you and go through the data and your questions, but I'll defer again to Michael and Robert when they might be able to show more of the slides.
Well, I would note that the slides are available on the Clene website. So if you go to www.clene.com, you can actually see a presentation by the two advisors and key Munich experts, Dr. Matthew Kiernan and Dr. Steve Vucic from the University of Sydney, who actually helped us implement the study. So there is a review of the data by them, and you can see the slides on the website if you just go to www.clene.com.
Oh, I didn't know that. So that's great news. So we'll put that in maybe in the chat for everybody, your website. So there's a question, maybe a little confusion about the results for limb onset. So maybe if you could repeat for the limb onset, did it help or not? It's somewhat confusing. Robert.
Yeah, again, for the limb onset people, which was 33 participants out of 45, we saw a statistically significant benefit at week 12 and a strong trend at week 36 with a p-value of less than 0.08. So yes, the limb onset patients were the patients that appeared to benefit with CNM weight just based on this peripheral electrophysiological measure.
Thank you. There's a question about whether it affects upper motor neurons. Is there any improvement on upper motor neuron dominant ALS patients?
Yeah, that's a great question. Michael, maybe you want to take that.
Certainly. So when we look at the rate of disease progression and we look at the overall population, including limb onset and bulbar, as an aggregate, so all patients that went into the study, and compare placebo to the CNM treated patients, the CNM treated patients had a lower rate of disease progression. And if we look at the subset and compare limb onset patients to bulbar patients, both of those subsets had consistent benefit. So we infer from that we're affecting both upper motor neurons and lower motor neurons. And then those data will be validated through the Healy platform study.
Yeah, I'm not sure, those are, I'm not sure you can quite say that to your point, Michael. I think you can say something in aggregate, but I think it would, because there's so much heterogeneity and the question about the upper motor neuron predominance, unless that's really, unless that group's really been shaken out, I guess I'd be a little hesitant to say that, but it doesn't mean, again, it doesn't mean it doesn't affect. I just don't know that we can say that more definitively.
And I think I'd agree with you. I get really cautious about the small numbers used in this study. I mean, there are some p-values here, but I think we should wait for the Healy data, especially given the larger sample size gives us more confidence with an appropriate powering.
Yeah, I would agree with that. In fact, I think if we really want to look at upper motor neuron, we would look at an FTD population, maybe. That's something we could think about in the future.
So there's a related question, like, what do we mean when we say statistically significant? And we're fortunate to have a statistician on the call. Laurie, do you want to explain that?
Sure. Always happy to help. So statistically significant really just means that the differences we see or the effect that we see, we are confident that that is a real effect, and it isn't just by chance that we happen to have these two groups that look different from each other. And that's really what we're looking for. We want to be confident that the differences we see are true, that they're true positives and not false positives and just by chance alone. And how we determine that is a more complex issue that we will have a webinar on, but very specifically, that's what it is. We want to be confident. Yeah.
You get more confidence with a larger sample size.
Larger sample size usually leads to more confidence, yeah, for a true effect. Yeah.
So there's two very nice comments from people who about the Clene EAP, just really thanking the Clene guys. That's for providing EAP access. They're very appreciative, and so are we, the community.
Thank you. We're trying to do our best for you.
There's a comment about, again, the sample size that with half on placebo, did that mean that about 22 or 23 got drug and the same number placebo? It was a 1-to-1. I mean, it was a 1-to-1.
Correct.
Yeah, great. Thank you. Okay. Let's see. There's again a thank you to the Regimen C for coming on. I think that is, I think that's all the questions here so far. Is there anything else that that either Robert or Michael you want to add, or Nick or James?
And I think we'd like to commend everyone's involvement in the Healy platform trial. It's the commitment to research that will help people with ALS in the long run. And research can be difficult and challenging. And we appreciate everything you do to support the HEALEY Platform Trial. And we thank you for Clene's participation as well.
Yeah, I would echo that. And I've said this before, and I'll say it again. What you guys have done at the Healy Center is astounding, really. I used to work at a very large Swiss-based corporation, and we would have had, 70 people working full time. And it seems to be, sort of your hobby here. It's your part-time job, and you guys have done a spectacular job. You built your own Oracle clinical database, and you've essentially you have your own CRO to do the study. It's pretty astounding, honestly, because I've been doing drug development a long time, and what you guys have done is you should really commend yourselves. It's an amazing effort.
Thank you, Robert. I really should end right on that high note, but there is another question, but thank you for that. It's a community doing this. So the question was really about how long it might take to see an effect. So are you seeing improvements or differences in the first weeks, and does the improvement continue longer, like in the open label?
Yeah, that's a great question. So far, we're doing several different kinds of studies with CNM-Au8 in different populations. So far, we have not seen a plateauing of effect through at least 48 weeks in any of our studies. So in all the studies we've done so far, the benefit of CNM-Au8 seems to accrue over time. And in the Rescue ALS study, it certainly accrued over time through week 36. And we continue to see what appears to be some perhaps survival benefit in the open label extension, but that's, of course, versus an external comparator group. So that has to be taken with caution. But the point is that I think given the pharmacokinetics and pharmacodynamics that we know of CNM-Au8, probably takes a few weeks for the drug to take effect, probably at least 12 weeks. And then we really do not see a plateauing effect over time. So that's what we've seen so far.
I think there's a good question. I think it's good to answer because I did see on the social media that there was a little bit of negative press, and I think maybe because some of the titles. And I personally was surprised because I was encouraged by your results. So the question is why do such a small study, and what can we learn from it, or what did we learn from it? And maybe that's a good question to end from Clene.
Sure, I can give you my perspective, and I think Michael actually was helped to design the study, so I think he's probably a better one to answer the question. But we're not a large pharma company; we have limited resources. We applied a grant to a foundation in Australia who agreed to fund the study, and we decided to do the study with this endpoint that had never been used before, because we felt it might be more sensitive and might be able to show the therapeutic benefit of CNM-Au8 in a smaller population. And that was the goal of the study. We learned a lot from this study. We learned how to use Munics and what populations to use Munics, at least in early ALS populations. So I think the study overall was, from our perspective, it was an astounding success. But again, Michael may want to comment.
Thank you, Robert. I think just as a general point in pharmaceutical development and biotech development, the purpose of a phase 2 study is to generally prove that the compound is safe, rather, to look at different possible doses and see if you have a sufficient efficacy signal to make the larger investment to do large studies. And that's how we designed the Rescue ALS study with the 45 subjects. And our interpretation of the data is that there's a sufficient signal there that we're really excited, and that if we weren't a partner in the HEALEY ALS Platform Trial, we'd be making that investment now and doing our own large study. But fortuitously, we think we have a signal now, and we'll be verifying that signal with the partners at HEALEY.
Yeah, so I think the science is as good as it was the day before you answered, but you did this result, and then this is kind of another piece of data. Excellent. So I think we're going to move to our biostatistician first lecture of a series, and I want to thank Laurie for coming and doing this and agreeing to shift it half an hour so we could talk to Robert and Michael and answer your questions. Thank you.
Thank you for inviting me. And oh, I am unmuted. I'm really been excited to be part of this study and this trial and the group here at MGH. And so as was said already once, it takes a village. It really is a very large effort. So I'm one of a large team of biostatisticians that are part of this study. On the left, you have the biostatisticians that are here at MGH, many of my colleagues. And then on the right, we have another group that specializes in Bayesian models, which is the model that we're using for our primary outcome. And at some point during all of these biostats webinars, we'll invite them to come as well and talk a bit about their Bayesian model and the primary analysis. So I want to actually thank the big group of biostatisticians that I work with. It's quite fun. We all come from very different backgrounds, which really blends together a lot of expertise to produce a really nice group. So thank you to that. And next slide. So as was mentioned, we're going to do an entire series of biostat webinars. We're starting here just very briefly. I'm going to introduce myself a bit and tell you a little bit about how, as a biostatistician, we think about research and how we're thinking about this particular study. We hope that this is the beginning of a series. We've learned over the time that it works a lot better if I talk for just a little bit and then have a lot of question and answer. So that's how we're gonna make these right now. So next slide. How we did this is I started by curating a lot of questions from both past webinars, Facebook sessions, Ask Me Anything sessions, emailed questions that now get forwarded to me and sit in the back of my brain as I do other things during the day, trying to come up with answers that work. So I'm not sure who is managing this, but can you click a few times? And here are some examples of some questions that have popped up. So we've gone through a lot of these questions. I've tried to gear different topics to hit a bunch of different questions. We can't do all of them in a half an hour. I think everybody would be very upset with me if I tried to rush through things. So we're just going to try to get them over the series of seminars. So next slide. So today's topic is really how to think like a biostatistician. So when I was first asked to do this and it first came up, I volunteered immediately because I love talking to people and I do a lot of teaching on many different levels in many different parts of the world. So I thought this would be a wonderful thing to do. And the more I started thinking about it, the more I started really just contemplating how I think about things. How, as a biostatistician, I think about research, I think about trials, and I think about numbers and people. And I realized quite quickly that I have a privilege as a biostatistician, and that privilege is that I don't think in terms of individuals, I think in terms of groups. My entire job and my entire profession is thinking in terms of groups. This group of people performs better than this group of people. If I give a drug to this group of people, they'll improve as opposed to this group of people. This is how statistics work, because it's the only way we can really understand that is this drug effective in the general population? And I've always thought that way because that's what I do, right? And I realized very quickly that it is a privilege. I don't have to think about the individual. I have to think about people in terms of a group. Whereas I know many people in the audience today don't have that privilege. And to them, every study is a sample size of one. And be it them, the sample size of themselves or someone that they care for. And that is very disconnected from the way that we talk in terms of reporting our results and in terms of thinking about our studies. And so I want to make a pact with you today and say, I will try very hard as I talk to think about what it means to hear what I say when you're thinking about a sample size of one. And in turn, I hope that you think about what I say and think about it in a way that, you know, what we're trying to do is decide if this drug will help a population of people. And in order to do that, we need to be able to think in groups. So if we both kind of keep open minds and realize that both of these opinions are important, I think that, as I said, my pact with you is that I never forget that most people that hear what I talk about here today and the results of our research are really thinking about a sample size of one. So I'm going to now talk you through the Healy ALS study as if you were a biostatistician. So next slide. One more. I'm sorry for the clicker, but you're gonna have to, I have these animated, which I'm realizing might not have been the best choice. So stop, perfect. So we're used to these generic, I'm calling them generic clinical trials. This is how you think of, and this is how a standard clinical trial is done. People get consented to a study. They're sent to either randomized into a drug or a placebo. The study continues. And at some point, the data is analyzed and there's possibly an open label extension as it goes on. When we do these studies, they do tend to be one-to-one because we need, our study is only as powerful as our largest sample size. So 100, in this case, I have 120 and 120, and you really only test one drug. And that's what historically we've been doing. Now, click, click, click a few times, please. Which means every time there's a new drug, you got to do another one of these studies, right? So you have 120 plus 120 in one study, 240 in the next, 240 in the next. And you can't share, and they go at different time points. And a lot of times, if you're in one study, you're excluded from another study. And although this does produce good data, this is not an efficient way to do clinical trials. And that's where this Healy platform study really comes in and shines. So next slide, and click like twice. Yeah, so I'm going to talk you through a little bit on how Healy is different from this. So people come in, they get consented into the platform is the terminology that we use. So inclusion, exclusion criteria, we believe that they make sure they understand what they're signing up for. All of this is unblinded, at which point they get randomized into one of many active regimens that we have. So click again. Right, so maybe they got randomized into A, B, or C. There's an equal chance that they're going to get randomized into one of them. This is also what's called unblinded. Everybody knows which one they're getting into. Statisticians are the ones that design these, we call them schedules as to when somebody gets sent to which one. We're the ones who monitor that A and B and C are all filling up at the same rate. And then click again. One more time, and then once they're in that study, they're consented in the study, and then they get randomized into a drug or a placebo. And you will notice here is different from the previous slide, where we have 120 in the drug, but only 40 in the placebo. And click a few more times for me, please. Okay, stop. And at that point, they're blinded. So you don't know if you're in drug or placebo, but the benefit of this platform is that those 40 in each of the placebo groups end up getting brought together so that we have 120 placebo to compare to 120 of drug A. Click a couple more times for me. After the, yes, perfect. And as more regimens start going in, one more time. Click, yeah. As regimens keep going in, people will get randomized into drug or placebo, and then that shared placebo group will grow. So we're never stuck with limited placebo groups where we have only 40 in one of the placebo groups. We actually end up having a very large placebo group while still being able to enroll many more people than we would have if we were doing separate studies for each of these drugs. Now, where is a biostatistician in all of this? Biostatisticians are sprinkled throughout this entire process. We have been at the table since design, since implementation. We think with the clinicians about what meaningful results are. We get advice from them about what's clinically meaningful. We in turn give them advice about how many people they need to see a significant result. We're involved in the randomization. We're involved in following through as the study goes and making sure the randomization is balanced, that we have balance across all the different groups. We're obviously involved in the analysis phase. We're involved in interim analyses to make sure that the safety is correct. We're involved in every aspect of this. And as I said at the beginning, we're always thinking in terms of groups. Are the people that are in regimen A similar to the people in regimen B? The same percentage of men, the same age ranges, the same types and different subtypes of the disease. We're always thinking about it in terms of that. We're looking at things like, Are the people in the drug group similar to the people in the placebo group on all of these different demographics? We're always watching these numbers as they go along. And then once the study is done and it becomes unblinded and we're able to look at the data all together, then we're very interested is, did the people on drug perform better than the people in the placebo group in all of the placebo groups? Okay, next slide. Okay, so that's really what I wanted to do. I wanted to give you a bit of an introduction and thinking a little bit about like how biostatisticians think. I also plan to introduce myself, which I realized I did not do adequately, so I apologize. I think I got excited. So my name is Laurie. I'm a biostatistician of many years. I spend my time split between doing research, neurology research, the ALS clinical trial, and some other things, and also teaching. So I teach a lot, both here at Harvard, but also internationally. And my favorite class to teach, and one that I'm very fond of, is something that has been titled Statistics for People Who Hate Math. If you have any children in college, I encourage you to take that class. So it's a wonderful class. But what I'd like to do now is open the floor to questions. I had closed the Q&A because I thought it might be a little distracting as I was talking, but I'm going to open it again. And is it okay if I go through the questions, or do you want to?
That would be great, Laurie. Thank you.
Okay. Okay. I'm going to go through, since A, B, and C are pretty much filled, how can we hear when the true findings are? I think that was left over a little bit from before, but you're right, A, B, and C are filled now. And as you know, the study follows people for 24 weeks. So once the last participant finishes their last visit at 24 weeks, we lock the data, at which point that's when we analyze the data. So we have a very tight time frame from the moment that the last patient has their last visit. And then as soon as we possibly can, usually within 2 weeks or less, we will have the initial results and then continue. And that's just the initial results of the study. The OLE, or the open label, will also be analyzing some of that data as it moves forward. Okay. How do dropouts affect the study count and results? Do you have a number in mind for allowable dropout that will still let statistics meet the sought after p-value? The sought after p-value, I like that phrase. We do take that into account. We use our information that we understand from previous studies, what percentage we saw drop out in previous studies to estimate. So we make a guess on how many people we think might drop out from this particular study. And that goes into our equations for the sample size that we need. So our sample size is 160. That 160 accounts for the fact that we expect a certain percent of people to drop out. So it isn't really 160 that we need at the end. It might be that we need only 150, but we're going to enroll 160 because we expect 10 people to drop out. So that's always taken into account. The higher treatment to placebo ratio is better for people in the study. In a single trial, are there particular numbers of participants needed to be able to have a greater than 1-to-1 placebo ratio? So 2-to-1, 3-to-1, etc. Every trial is a little bit different. I like to talk about how our power is determined by our smallest group. So if we do 3-to-1 or 2-to-1, the bottom line is that that small group is the number that really tells us how powerful the study is. Power is a funny concept. It really means just the probability that we're going to see a significant result if that result actually truly exists. So there is not a particular number. Are there particular numbers of participants needed to be able to have a great? No, it really depends. As I said, I think it really just depends on that one number of the smallest group. So you can enroll more actives as long as your placebo group is big enough, which again is why the Healy study is so great, because we get to combine those placebo groups.
Perhaps, Laurie, in that question, I just wanted to ask you, can you confirm that for a certain number of people, the highest power is if you have one-to-one randomization. So if you were to change the ratio, you would need to increase the sample size, correct?
You would need to increase the sample size. 1-to-1 gives you the highest power because it's that one. So 2-to-1 will most likely have a similar power to 1-to-1 because it's that smaller group that we care about in terms of power. Okay. So what does p-value mean? Love the question. I have an entire talk that we won't do here, but we can do bits and pieces where I explain a p-value in like 17 different ways. Because one of them will find understanding with everybody, right? So a p-value in very simple terms, because it's correct, is the probability that the result that we found is real and not just by chance. That's all it is. So when we say we want a p-value of less than 0.05, 0.05 means, given our results, we think there's a 5% chance that we just happen to find the right sample of people to see these results, but that if we did it somewhere else and we ran the study again, we wouldn't see those same results, right? So we believe that there's only a 5% probability that the results we found are just by luck. And we've sort of kind of agreed as a society that a 5% false positive rate is real. That's what it's called, false positive, right? That a 5% rate is kind of the one that we like to talk about. Is it greater than 5% or less than 5%? So it's the probability that what we saw was just luck and didn't have anything to do with the drug. Platform trial is very efficient use of patient resources, such as placebos, allowing for more patients on active drug. How do you determine when to start a new placebo pool and what groups to include? That's an excellent question. We will be using those historical placebos as we move forward, but we're always discussing whether the placebos that we've enrolled over the last year are the appropriate population to compare to people who are taking drugs, say this year, next year, and the next year. It's easy to think about when we think about, people who are on active treatment this year, comparing them to the placebos since the beginning of the study seems very logical. But if we think maybe 5 years in the future and, hopefully these drugs would be effective, that the standard of care would have changed, it might not be appropriate 5 years from now to compare it back to placebos that we had from this year. And we would need to make sure that our placebos have the standard of care that is 5 years from now. So we're always thinking and making sure that those placebos are the appropriate comparator group to our active treatment. What they teach in class, which means what we ideally would love to do, but we are never able to achieve, is that your placebo group should be identical to your active drug group with one exception, and that is taking the drug. I'm going to jump around a tiny bit just to keep on the statistics theory, the statistics theme. So will people on OLE find out whether they are on real drug once the last person enrolled and the stat workup is done, or do they have to wait until all people who decided to do OLE? So once the last person is enrolled and finished their 24 weeks, then we are unblinding it and people will be able to learn what they were on before OLE. So once everybody finishes their 24 weeks and determines whether or not they want to go into OLE, everything will be analyzed and it will be unlocked.
However, Laurie, for the purposes of from the participant perspective, so we're going to be able to let people know if they were on active or placebo, only later when basically the OLE ends as well, because the OLE is also part of the trial itself. So just wanted to kind of again look at the participant perspective versus study team perspective. Yeah.
Does the randomization process attempt to make the regimen pools uniformly heterogeneous? Which is one of my favorite words to say, by the way. Age, sex, all that sort of stuff, right? Yes, that's the magic of randomization. Randomization isn't magic, but that is the magic of randomization. It is, we are able to make these two groups as identical as possible for both known things that we measure, such as age, sex, time of onset, different subtypes, but also, and that's what's magic about it, is unknown things. So things that we haven't measured that we might not know are related. Those also make those two. Randomization allows us the confidence that the two groups are equal on those as well. What is the target p-value? I just lost that. Less than 0.05 for general, especially FDA accepted as statistical significance. So wonderful question. That is a question that will be continuously debated throughout the ages, and I have to say also has been very strongly debated in the literature in the last few years. It's one of those things that it's been that way. We feel comfortable with the 5%. Sometimes it's actually 2.5%, depending on how you do the analysis. But I can't honestly say that there is anything special about the number 5%. I actually would invite other panelists here to weigh in. As I said, the 5% seems to make us feel comfortable of like, I'm okay with a 5% chance that I'm saying that this drug works when in fact it doesn't work. But it really is something that's been heavily debated in scientific literature lately. I don't know if other panelists have thoughts, Sabrina.
Yeah, I mean, real quick, I think that, numbers are numbers, but there's also the totality of the evidence, meaning that you also want to look at other parts of the trial, other trial results, safety, secondary outcomes, etc. So it's really never just one number, but that particular number in conjunction with all the rest of the information that the trial provides. Yeah. So I think that's important to note. And Laurie, I'm sorry, because this is so engaging and so fantastic, we're getting an incredible, we're getting so many questions. I'm not really sure how to take all of them. I wonder, in the past, what we have done was to try to maybe do some rapid fire so that we address all of them to make sure that people get an answer now. But I do want to point out that you will be back on December 16th, which is another...
whatever that Thursday is, yeah.
Yeah, the 16th, yeah. So I think it's actually the last webinar of the year for us. So we really end on a high note with you. So perhaps maybe you can take a few questions sort of rapid fire.
All right, I'm going to do rapid fire, which is not my usual. So I see a couple of questions about where the FDA stands with this. I think there was at least 2 of them. We do consult with the FDA. They want to know everything that we're doing, and then they want to fight with us about our decisions. And by fight, I mean we have long discussions to come up with the most appropriate answers. So there is quite regular feedback back and forth. How, with N of 160, if the subgroups can be analyzed with significant confidence? The N of 160 was determined for the primary analysis and not the subgroup analysis. The subgroup analysis will be what's called underpowered. It doesn't mean we won't find stuff in them, but we did not design the study to find stuff in them. And there are other idiosyncrasies with subgroup analysis, which we can talk about in another one. Might the placebo group require fewer and fewer people in the future? I think that is not a bad question. And I think that that's a question that's open to discussion. And it is something that we're always contemplating. Can you explain the difference between intent to treat and modified intent to treat? The Torsen data that was reported used MITT for fast progressors but non-MITT for slow progressors. Do you know what the difference between, every time something happens, it moves, versus the total ITT group? I would like to get back to you on that question, if that's okay, just because I want to look at the data and make sure I completely understand what they did before I say anything incorrect. So I'm more than happy if someone has the email to go back to that question, if that's okay. How do you account for effects of other medications on outcomes when not everyone is taking the same cocktail? Just as an example, with folks on Radicava, some folks are not on it. That's true. It goes back to the randomization. We do take that into account on some of our analyses, but it also goes back to the randomization, where the randomization will help ensure that we have an equal number in both groups that are taking the different cocktails. I will also say that based on our consultation with the FDA, we are stratifying our randomization by the 2 drugs that many people take. So we are ensuring that the distribution of those 2 drugs are the same for all the different regimens and then within the regimens as well. And that was some feedback from the FDA. In other words, are you looking for 95% confidence? Yes. 95% confidence is the same thing as saying P less than 0.05. Yes. A drug that gets approved in the middle of the Healy study, it will become standard of care. What's the statistical profile? Isn't it important to preclude trial participants from taking a new approved drug so that it will not skew the outcomes? Okay. Sabrina, can I can talk about this from a statistical perspective, but not a clinical one.
Yeah. Basically, the question is what happens if AMX35 gets approved in the middle of the Healy trial? As we discussed in this webinar before, we have a commitment to allowing people to take standard of care medication. So as of right now, that's Riluzole and Radicava, and we stratify for them, but we allow people to make their choice, and we still keep the groups balanced by using, again, the design features that such as stratification. And then if AMX35 were approved, then we would have, again, we would want to allow that. The exact timing or logistics of that obviously are we haven't worked them out because there's many unknowns. We don't know exactly when the drug will be approved, etc. So we are definitely gonna, obviously we follow that really closely, and I'm sure that we will have mechanisms to allow that. But again, that's gonna be discussed when we get closer.
Yeah, and I would say that there's also, the likelihood that the people that want to start that drug are going to be evenly distributed across the group. So, a cardinal rule in terms of statistics and research is if you don't collect the information, you can't analyze the information. So we will obviously collect the information on whether or not people are taking that drug, at which point we can incorporate it into our analyses. Okay. Thank you. Did I do?
Yeah, I do want to end with a comment. Somebody said, not a question, but Dr. Chibnik is awesome, and I look forward to hearing more from her. So we agree with that. I would suggest that, we know you're coming back on December 16th, and I'm sure in the new year we will have you back again. But for December 16th, if you have any questions, please feel free to send them to Catherine and Alison, our patient navigators. So the type of talk and all of these really is based on your feedback. So we can certainly plan around what matters to you, what questions you have. If you feel like, some of the questions were not clear or answers were not clear, or you want to learn more about a specific topic, please send us your questions, and we'll make sure to start with those next time. And next week, we're not going to be meeting because of Veterans Day. And so we will be meeting the following week for this weekly series. Thank you so much.
Thanks.
Thanks, everyone.
Thanks, everyone.
Bye. Thank you.